Our speaker today is Magalska. She is a member of our silviculture staff here at the DNR.
Thanks, Ralph. As Ralph said, I've been working in the silviculture department, so I'm in the Forest Resources Division of the Washington Department of Natural Resources, and I have been working in Douglas Fir tree improvement in the Pacific Northwest for about 8 years now. So I'm going to be talking today about some of the work that I've done with the Tree Improvement Research Cooperative at Oregon State University. I wanted to start today by going over a couple of forest genetics concepts that I thought might be helpful when we start digging a little bit more into some of these research topics, and then also go over a brief history of Douglas fir tree improvement in the Pacific Northwest. Then from there we'll get into the co-op's project background and dive into the 4 different components of that research project. So I wanted to start off with a few forest genetics concepts today, and I wanted to give credit to Keith Jayawurkama and Terrence Yee at the Northwest Tree Improvement Cooperative. They have put together these slides, and I am presenting some of the information that they have previously put together. So as many folks I'm sure know, when we're looking at any sort of organism, any sort of plant, animal, person out there, we're looking at its phenotype, and the phenotype is composed of both the genotype and the environment. But we're not really, when we're working in tree improvement, we're not really interested in the absolute values of the phenotype or the absolute codes of the genotype. What we're interested in is the variation of both of these things. And in particular, when we're working in tree improvement, we're interested in the genotypic variation. So to get into genetic variation more specifically, there's a few different levels of genetic variation that we're interested in, and they get a little bit more specific as we go. So starting off, when we're talking about a species may have geographic variation of its genetics. And that means that with the different populations, we're going to see different gene pools. And then if we think of these 3 trees down here as each one of them being representative of a different family, we have an example of among family genetic variation. But if each of these 3 trees were part of the same family, then what we would be looking at would be within family genetic variation. And finally, we can also look at within tree variation. And a good example of within tree variation would be wood stiffness properties. Wood stiffness properties can change throughout a tree, and they also change over a tree's lifetime. It's often common that juvenile wood properties are a little bit less dense than the mature wood, and so that's just an example of within tree variation. So once we're interested in the genetic variation and we have all of that information, we've tested that information, we're using the genetic variation to estimate some other factors about our population. And one of the things that we're most interested in is how heritable a trait is. It means how can this trait be passed on from its parents to its offspring. And when we're talking about heritability, we're specifically talking about the proportion of phenotypic variation that is due to genetic rather than environmental factors. And if you look at the second bullet point, it's the equation that I showed you earlier, where we had phenotypic variation on one side and it equaled genetic and environmental variation on the other side. Well, in this case, we're dividing the genetic variance by the phenotypic variance, and that tells us how heritable this particular trait is. And when we're talking about a specific trait, we might say its heritability is 0.4, and in that case, we're saying that approximately 40% of the variation that we're seeing is due to the genetic factors, and 60% of the variation is due to other factors, most likely environmental factors. When we're working with Douglas fir, most of the growth traits, these being height and diameter traits, are usually under moderate to strong genetic control. So they have heritabilities ranging from 0.2 to 0.5 when you're looking at a single site, or when you're going across multiple sites, you can have up to 0.3. The higher the heritability is, the stronger that trait is under genetic control, and the more you can do with it in a tree improvement program. Once we have the heritability, we can estimate the genetic gain. When we estimate the genetic gain using the heritability and the selection differential. The selection differential is the difference between the population that you started with and the parents of the population that you're going to have. So if all of these trees are part of our original population and we're selecting for the best height growth, we would just select these darker green trees here. And so we would take the average of these darker green trees and compare that to the average of all of these trees, and that would give us our selection differential. And the selection differential is usually given in the percent of the mean over the original population. And this starts to get a little bit confusing because in some traits we're not always necessarily trying to improve them by making them by getting more of them. You know, particularly when we're talking about stem defects, we want our genetic gains to be negative. In addition to looking at traits individually, we also like to think about traits of a tree together, or traits of populations together. And sometimes we have phenotypic traits that are varying together along some continuum. And in these cases, the traits could either be controlled by the same set of genes, or they could be controlled by genes that are close together on a chromosome. When we're dealing with genetic correlation, it means that we can have either intended or unintended correlated response. So if we're selecting in our program only for height, we're still going to have an effect on the different traits that are genetically correlated with height. So if we select for the fastest growing trees on height, we'll probably get trees that also have fast diameter growth, but we might also get trees that second flush or burst bud too early in the spring. So you have to look at the correlated response of some of the other traits sometimes when you're working in a tree improvement program. So that brings us to a brief history of tree improvement in the Pacific Northwest. And again, I wanted to give credit to the folks that originally put these slides together. It's Nancy Mandel, who I don't believe is with the Forest Service anymore, but was a big part of tree improvement in the early days, and then Keith and Terrence down at NWTIC again. So trees are just like any other agricultural crop. They can be bred and improved just like corn or chickens can be. And globally, tree improvement started back in the 1950s, right around the time that there was a pretty big housing boom with rapidly growing economies. And it also was a time where people were fairly confident that forest trees were under genetic control and that these trees could actually be improved. So tree improvement in the Pacific Northwest specifically got started in the late 50s and the early 60s, and oftentimes this started with the first grafted orchards. You can see a grafted orchard out here on the Olympic Peninsula. The Denial seed orchard is one of the earliest orchards. And the primary goal of the seed orchards that were established were to have seed of a known geographic origin. But these were typically untested and phenotypically selected trees. And oftentimes another way to refer to phenotypically selected trees is to just call them woods run trees. But in the early 60s, the Pacific Northwest Research Station decided that we could do better than just select woods run trees. We could actually breed these trees, assuming that we would also be able to maintain local adaptation. And so Roy Seiling put together a proposal, and given that tree improvement with Douglas Fir is still going on in the Pacific Northwest, I think it's safe to say that it was well accepted, and industry and agency decided to get together and cooperate on a few different tree improvement programs. So I wanted to talk a little bit about the tree improvement cycle. So as I previously mentioned, woods run trees. So that's where we start with a selection of our parent trees. And the woods run trees for the first cycle of Douglas fir breeding were typically 20 to 30 years old. They had good stem form, were either dominant or co-dominant in their stand. But they also had to have good scion, good branch tips, and they also had to have a cone crop, because if they weren't reproducing, you can't bring them into your breeding program. So you would collect the scion and the seed from these parent trees, and you would establish a clone bank and a breeding orchard. And then we would also establish progeny tests. So we're taking the open pollinated seed that was collected from these parents and planting those out in progeny tests. And at progeny tests, we have the same families planted at all the sites, and that's important for the work that I've been doing. From the progeny tests, we can select the best parents again, then we start the process over, we collect and graft scion from those best parents, start breeding, do more progeny tests. So right now in Oregon and Washington, the second generation of Douglas fir and tree improvement is about wrapping up, and we're moving ahead with third cycle improvement. And so we're in the breeding phase and starting to move towards establishing progeny test sites again. The tree improvement cycle isn't valuable if it doesn't move into the production or the operational side of things. And so once parents are selected, seed orchards are established, and those seeds are processed, the cones are processed, and we end up with the seedlings, and these are the trees that you would pick up from Webster Nursery. So there are a lot of first cycle tree improvement programs when it first started off. That you can see those on the left. So those are the different breeding zones, and then on the right are some of the test sites. A very, very large number of parents were selected and tested across the region. So over 30,000 Douglas fir open pollinated families were tested. About 3 trees were selected per 1,000 acres of timberland, and this resulted in around 1,000 test sites, 3.5 million progeny tested, and 9,600 acres of plantation. So this was a really, really big project, and it was done cooperatively because it was a big project for any one person to take on by themselves. So different organizations of private companies and public organizations got together and worked on different programs for their different land bases. What's important about these different tree improvement programs is they give us the ability to assess family and individual trait heritabilities. We can look at the genetic correlations of different traits within these families, and then we can also start to think about what the effects of artificial selection are. So what kind of genetic gains are we getting? What are our correlated responses? And then there's a lot of data associated with these test sites. So each program had up to 3 different measurements for height and diameter, and many programs also had different stem form measurements done. So that gives me to the Pacific Northwest Tree Improvement Research Cooperative project background. Why would we want to use these progeny tests? So walking across the landscape, you come across stands and you see stands that are more productive than others. And the differences in the productivity can be both environmental and genetic, but in most places these differences are confounded. But we're really only interested in figuring out what some of the environmental differences are in influencing the productivity of these sites. So we can use these progeny tests within these different breeding programs to try to remove some of these genetic differences and basically try to look at what the productivity would be across the landscape if the exact same tree was planted at all of these different spots. So at a higher project level, one of the things that Glenn Howell has been trying to do is to come up with different ways to model a site. So you can characterize a site as having high frost damage or high drought risk or high fire risk or low fire risk, and you take all of these different measures together and you come up with an idea of how well that stand is going to do over its lifetime. And so we can use this information and feed it into mechanistic growth models and make a variety of decision support tools for land managers in terms of what kind of trees they are deploying across their landscape. I'm sure many of you are aware that there are currently a lot of different forest management models out there, and one of the ways that you can determine how volume growth of your stand is using an empirical growth and yield model. And this requires, standing trees. We're basically predicting future performance off of past data. And that can be problematic because we're not taking dynamic site characteristics into consideration. On the other side of things, we could use a mechanistic model. And in a lot of cases, the outputs of these mechanistic models are like net primary productivity or leaf area index, which isn't necessarily valuable for someone who is trying to put up a timber sale. That's not good information to have in order to auction off the sale. but it is dynamic. It does change with changing environmental characteristics. So what is a way that we could look at site productivity and try to combine the best aspects of both of these different types of modeling? And that's one of the things that we're trying to do with the site characterization project. We also wanted to take a look at stem form. It is not a trait that has been looked at very thoroughly in the past across large landscapes. We're interested in two different types of stem defects. One is the fork, which I'm sure most people could see, but one that is a little bit less commonly known is the ramicorn branch, which is a large, steep-angled branch, and this one I think is actually a better example because you can see it kind of starting to wrap around the bowl, and that can create all kinds of problems in the wood quality. It causes stem deflection, makes a really big spike knot, and it just can really decrease the value of your log for quite a distance from where the defect is. So we want to have a better understanding of what sort of site characteristics are influencing stem form and making some of these undesired traits come out in our plantations. And like I said, stem form does have an impact on tree value. There was a case study done a little while ago that showed positive genetic and environmental correlations between growth and frequency of forks and ramicorn branches. So we were seeing more stem defects on the faster growing plantation. But across the Pacific Northwest, the relative roles of the genotype and environment were still a little unclear. And a lot of foresters were coming to us and saying that they were seeing an increase of stem defects associated with rapid growth and the proximity to the coast. So we're also interested in kind of unraveling these genotype by environment interactions. And as I mentioned before, when you're working with progeny tests, you have the same genetic material planted on all of these tests. And so the variation that you're observing among the site productivity and the stem quality can be attributed to the environment. And the ability to pull apart this G by E is what makes our project unique among other site characterization studies. And finally, we're interested in site characteristics because we want to be able to dynamically model how Douglas fir might respond to climate change. In the Pacific Northwest, the projections are a little bit uncertain. There's uncertain changes to precipitation. Summer warming will be more pronounced inland, but greater winter warming than summer warming in the Western Cascades. We'll also see increased carbon dioxide. And our questions for all of these changes will, how will Douglas fir respond, and which climate site characteristics should we be looking at in the future in order to, again, deploy the correct genotypes across our landscape. And, in the Pacific Northwest, we're interested in what Douglas fir is going to do in the future because it's an economically important species for both Oregon and Washington. We have competitive markets, dealing with low-cost imports and people converting forested land to other uses. So really being able to understand these relationships is going to help the industry stay competitive going into the future. So we wanted to start off with a little bit of a pilot study and see if we had our methods well understood so that we could start to make some sense of all of these different breeding programs that we were looking at. So we started off with 191 different progeny test sites in Oregon and Washington, and these were located all west of the Cascade Crest. They were measured between 1967 and 2005, and they had a wide variety of elevation and number of trees planted at each of these sites. So then we wanted to figure out which site characteristics we were going to use to look at the variation among these sites. We started with Climate WNA, which is a climate modeling program put together by the folks up at UBC. We also used the NRCS Surgo data set. And finally, we used the USGS digital elevation model to look at 2 different site characteristics. So in total, we had 40 different site characteristics that we wanted to take a look at. And we combined that with our response data. And so we were looking at different periods of growth for diameter and height, and then we were also looking at stem forking and ramicorn branches. So we ended up with 7 different response data sets with 24 different response variables in them, and then our explanatory variables are site characteristics. And then because we want to try to remove the genetic differences across the landscape, what we did was take this across program data set, and make a within-program data set. And so we removed the variation that could be explained by the program and the sowing year. We took both the across and the within-program data sets and we performed a few different analyses on them. First, when we were starting with 40 different site characteristics, we needed a way to group them together so we could better understand the results that we were seeing. So we did a hierarchical clustering of those site characteristics, and it resulted in, I think, about 5 different groups. And then we did 3 different variable selection procedures. One was just a simple correlation, so we were looking at one response and one site characteristic, and then we did a linear regression with one response and multiple site characteristics, and then a random forest model, which is also one response and multiple site characteristics. So we had those 3 different responses, the correlation, the random forest, and the linear regression, and we used the results of all 3 of those models to come up with a total importance score for both the across and within data sets. So the total importance score was kind of a measure of how consistent was this site characteristic explaining variation in either growth or stem form. And these are the results for our growth response groups. You can start to see a few patterns come out here. Temperature 2 is comprised mostly of cold season temperatures, so you have minimum coldest month temperature and extreme minimum temperature in this group, and you can see that you're getting a lot of pretty strong importance scores in both the across program and then in the within program data sets. And we could also see available water capacity come out as being important over here, so we can start to see a few patterns for the growth responses. So our conclusions from that portion of the project, the pilot study, were that cold season temperatures and available water capacity seemed to be important to site variation. We were able to identify some of these general relationships, but it was still fuzzier than we wanted it to be. And so we started to think about ways that we could refine the analytical approach. And in particular, we wanted to reduce the number of independent variables that we were looking at within each of our models. So I'm going to take a pause on the growth aspects, and now we're going to switch over to talking about stem form. So for the stem form results, there's not a lot going on here. So there really weren't any site characteristics that were important for explaining the variation that we were seeing stem form across these sites. So we decided to maybe take a step back. There was very little variation explained. There was a lack of consistency across the different analytical techniques. So we're like, maybe we're looking at the wrong site characteristics, or maybe we just don't understand our assumptions about stem form in general. So we decided to do a study just on Douglas fir stem defects. And here we wanted to look at some of the general assumptions that we held going into the site characterization project and to see if they actually held true. So we wanted to take a look at whether or not faster growing sites actually do have more stem form defects. If those faster growing sites can explain the differences in the variation that we're seeing, whether or not there is a relationship between stem form and the proximity to coast, and then we wanted to have a better understanding of how our growth traits may be related to our stem defect traits. So we again used the Northwest Tree Improvement Cooperative data. There was 22 first-generation breeding programs that had stem form recorded, and depending on which stem form trait we were working with or which growth response trait we were working with, we had anywhere from 93 to 129 locations. So for the stem defects, we were looking at the average incidence per tree per year of stem forks. It's kind of fuzzy, but because these plantations were measured at different ages, we had to have some way to normalize them. A plantation that was measured at an older age had more opportunity to develop stem defects than a younger plantation, and we wanted to try to even that out. And then we made sure that any of the growth traits we used were taken at the same measurement age as the stem form traits. So we took a look at 6 different relationships to see if faster growing plantations actually do have more stem defects. And the answer is yes. We do see more stem forks and ramicorn branches with increasing height. So particularly with height, we have an R-squared value of 0.54, so that's pretty good R-squared value. We're seeing lots of the ramicorn branching explained by height growth. So for every additional centimeter of height growth you had in a given year, you could expect to see a 5% increase in the frequency of forking on your plantation, or a 6% and a 6% increase of the frequency of ramicorn branching on your plantation. The relationship also existed between diameter growth and stem defects, though it was not as strong, which is to be expected because other studies have shown that height growth is strongly correlated with stem defects. So the R-squared values are a little bit lower, but the response is a little bit stronger. So for every additional millimeter of diameter growth you have in a given year, you can expect to see the frequency of stem forks increase by 40% and the frequency of ramicorn branching increase by 50%. When we combined height and diameter to come up with volume, though, we did not see any variation explained or by the volume growth. And so this was inconsistent with the original analyses, but this is an analysis across 22 programs instead of across one program, and as I'll show you in some later slides, it would just kind of, we were either lucky or unlucky in that the pilot study happened to be the program with some of the strongest genetic correlations. So the other part of this was, are we seeing more stem defects near the coast? And again, the answer with that is yes. For every 10 kilometers you go further inland, you can expect to see your incidence of stem forks decrease by 15%, and you can expect to see the frequency of your ramicorn branches to decrease by 12%. So then we wanted to take a look at the heritabilities of the 2 stem form traits that we were looking at, forks and ramicorns, and they are low, but they are variable. So in some cases we have, programs that have, almost a 0.3 heritability for ramicorn branches, but they're mostly lower than that. So they're under less genetic control than the growth traits are. And as I mentioned, the pilot study happened to be the ones with the highest heritability for our ramicorn branches. And we also did take a look at whether or not stem defects and growth are genetically correlated, and again the answer is yes, they are genetically correlated, and it is mostly a positive correlation, but it is also important to point out that we do see some negative correlations, which in this case is a good thing because we don't want to breed more stem defects into our trees, we want to decrease it. So seeing these negative correlations in here is good news. So then we wanted to take a look at what would happen if we were to select solely on stem form, and then we also wanted to look at what would happen to our growth traits if we were just selecting on stem form and not paying attention to our growth traits. And so if we were to select solely on stem form, we could, on average, reduce the frequency of forks and ramicorn, or forks by anywhere from 3% to a little over 4%, depending on our selection intensity. And we could reduce our ramicorn branching by anywhere from 6% to just over 9%, again, selecting, on average, depending on our selection intensity. And as you can see, if we were just trying to minimize our stem defects, we wouldn't have that big of an impact on growth. It's important to note that we can choose to reduce the number of stem defects but still get modest growth gains, which, at the end of the day, volume is always going to be the most important trait. But if you're selecting on something else and you're not going to hurt your volume by doing that, that's always good news. And we also have our correlation breakers, which are good news too, where we can really really decrease the number of stem defects that we're seeing and continue to increase our volume growth at a rate that we would like to see in a program. So our conclusions from that project were that we are seeing increasing stem defect with increasing growth, and you do see more stem defect near the coast than you do inland. These stem defects are heritable, so the parents of the trees that you're planting matter. And typically we're seeing positive genetic correlations between our stem defects and growth, which is bad, but there are correlation breakers and variability among the different programs and sets that we're working with. So selecting solely for growth has a small potential to increase stem defects, but it is possible to select for growth and fewer stem defects at the same time. So going forward in a tree improvement program, we would want to continue to explore what sort of environmental characteristics are contributing to these stem defects. And we would want to include a stem form as a selection criteria in our operational breeding programs. And as our breeding programs move forward, to continue to monitor what type of relationship we have between these stem defects and our growth traits, so we don't accidentally breed more stem defects into a later population than we have right now. So that kind of wrapped up our stem form project, and at that point we were ready to step back into trying to figure out which site characteristics really explain variation in growth. And so as I mentioned, we wanted to refine our analytical approach, and so we did that by increasing the number of sites that we had. We wanted to focus on fewer responses, so fewer different growth traits. More climate WNA was expanded since we first started this project, so there are more climate variables available to us. We also added geomorphometric variables. We had to make the decision to remove the soil site characteristics because there were some problems that we ran into with the NRCS data that we just couldn't quite get past. And then there are a few papers that came out that had different analytical techniques in there that were very applicable to our project. And so we were able to improve both our random forest and our linear regression approaches. So in my original pilot study, we had 191 test sites and 22 programs, and we were able to expand that to 348 test sites in 32 different breeding programs. So that made a really big difference. But these are all still in Oregon and Washington, west of the Cascade Crest, and they are still measured between 1967 and 2005. For the site characteristics, as I mentioned, we got rid of soil, so we were only dealing with climate and geomorphometry. But you may recall that I mentioned that we wanted to reduce the number of site characteristics we were looking at, but the first study had 40, and now we have 90. So we started off going in the wrong direction, but that forced us to come up with some ways to better reduce these site characteristics. So we did that by just doing simple correlations among all of these site characteristics and removing ones that were highly correlated with many others. And then we used a principal component analysis of the growth and survival traits to figure out which site characteristics explained the most variation. And with a principal component analysis, what you do is you take all, in this case, 7 of our response traits, and each of those response traits have variation associated with them. But instead of having that variation associated with each of the individual response traits, we try to pack all of that variation into these principal components. And in this case, we had one principal component that represented growth and another principal component that represented survival. And in total, these explained 71% of the differences we were seeing in growth and survival across the sites. So then we took these principal component responses and we used them in a random forest analysis. So we identified the site characteristics that came out as being important in both models, and then we added back in a couple of a priori site characteristics to facilitate comparison to other studies. This included things like mean annual temperature or mean warmest month temperature, variables that other researchers have looked at in the past that we want to be able to assess how our project may compare or contrast to theirs. So we came up with a list of 19 candidate site characteristics that we want to use for our final modeling to try and predict growth at sites without having, trees there to measure or changing environmental conditions going into the future. So these are the list of the variables that we were starting with. We had 17 climate variables and 2 geomorphometry variables. And we had mostly annual variables, but we did have some seasonal variables. So from here, we stuck with the three-pronged approach that we started off with in our pilot study, but we refined it a little bit. So we stuck with the random forest, we stuck with the lasso regression, but instead of using simple correlations, we replaced that by using multiple imputation and multivariate regression trees and lasso regression. So the imputation, what that does is it fills out the data set. Not all of these programs were measured at the same age, and not all of these programs had the same traits measured each time they were measured. But if you want to do multivariate analysis on the response side, you need to have a complete set of responses across all of your sites, and we didn't have that. So we used the imputation in order to fill that out and see, you know, if the multivariate responses were useful as well. And so then we had these three different approaches in digging into how to compare the importance across different analytical techniques. We really learned that you can't do that, and there wasn't really a statistic or anything we could use. It was just as simple as whether or not the site characteristic was in or out of the model. And so we had to come up with some rules to try to determine whether or not the number of models that it was in was actually valuable and made it a site characteristic we would continue to be interested in. So these are just some of the rules that we came up with. They were loosely the same for our growth and survival traits, but because we had more growth traits than we had survival traits, they had to be a little bit different. So we ended up with this final table. The darker the row is when it's highlighted, the more important the site characteristic was. And then when we were getting down into it, we realized that there were a couple of different final models that we wanted to be able to compare to one another to really see which site characteristic was more valuable than the other. And so we wanted to know whether or not we can include the seasonal number of frost-free day variables or just the annual number of frost-free days. You know, did it matter if we used one or the other? Was the average spring temperature comparable to the mean annual temperature? Was it better to use extreme minimum temperature or mean cold month temperature? Was it better to use continentality or temperature differential in the model, or to just leave it out? And then finally, was it better to use the seasonal and evaporative reference or just the annual evaporative reference? So the final model that we came up with had 9 site characteristics in it. So it included 8 climate characteristics and then dissection, which is one of the geomorphometry variables. So then we took this final model, and this final model is something that we were hoping we could use to predict any growth trait. So we could predict volume, we could predict height, we could predict diameter, we could predict it at different ages, and it didn't matter whether or not that response trait was actually used in the development of that model. And so what we wanted to do was compare the best model for an individual growth trait. So if you developed a model just using that growth trait, how good of performance could you get, versus if you use this model that we came up with that has 9 variables in it. And so that's what this table is summarizing. And we used height, diameter, and survival to develop the model, but we did not use volume or stand level volume. So these are all individual tree models or responses, but this volume per hectare is a stand level characteristic. So as you can see in both the within and the across program data sets, the final model performed pretty close to the best model for some individual traits. So, you know, we had 0.2 r squared or 0.28 r squared for height using our final model, but 0.29 using the best model. And you can see some similar things here. If volume, one that wasn't even used to develop the model, 0.28 in the best, and then 0.33 in, 0.28 in the final and 0.33 in the best model. So, these aren't R-squared values that are going to break the bank or anything like that, but that they're explaining enough of the variability that we see in productivity that we feel that these are site characteristics that we need to be paying attention to. And the difference between the R-squareds and these models is low enough that we feel confident moving forward with this final model to predict different growth responses, if that's what we want to do at different ages. So as I said, the final random forest model was performing well for the individual traits when compared to their respective best models, and it performed well both across and within groups. So it was performing both regionally and locally. So now what do we do with this? So the annual variables performed as well as the seasonal variables, and we were seeing interactions between temperature and precipitation. Those come out in the annual heat moisture and the evaporative reference. Extreme temperatures are important, so how cold does it get in the winter and how hot does it get in the summer? And growing season length is also important. Precipitation as snow is coming out is important, but we weren't really sure if this is a water availability or a temperature indicator. And then the terrain of our landscape seems to be influencing the interactions of these temperatures and precipitation. And so one of the outputs that random forest models gives you is a variable importance measure. And so these are the variable importance measures for the across and the within program data sets. And for stand volume at measurement 3. And in the top 3 for both, we see precipitation as snow and annual heat moisture index. So it's the top 2 over here, and it's the top 1 and 3 over here. And again, this is, predictions for a response trait that was not used to develop the model. And so these are what's coming up as important when we're predicting growth of a site using just site characteristics, not using any prior knowledge of how well trees have grown there in the past. So we want to be able to use this information to better understand what the effects of near-term climate change may be on Douglas fir productivity across the region. And I do have to mention that this portion of the presentation is still, the results are still in the preliminary phase, so things might change between now and when we hopefully get the manuscript out. But there's still results that I think are very interesting and very close to what would be in the final paper. So we decided to look at 3 different future climate periods, and Climate WNA allows us to look at 3 periods, 2025, 2055, and 2085. And these use 15 different climate models combined. The folks up at University of Alberta made an ensemble of all these climate models, so you don't have to get into which climate model are you using and which general circulation model. It was just kind of an ensemble. It was a mean of all of these, and so it was more robust. But we still looked at 2 different emission scenarios. We looked at RCP 4.5 and RCP 8.5. And we continued to predict how stand volume per hectare, so it's cubic meters per hectare. And we wanted to look at both the plantation, so the individual progeny site level. We also wanted to see if we could see any regional patterns. So this is looking at the individual sites. And each of these lines, I know there's a lot of them, each of these lines represents one of those 32 groups that we looked at. And so this is the across program data set. So this is with genetics and environment still confounded. And you can see in both emission scenarios that most of these lines are just tracking up. And so what this is telling us is that we could just expect to see productivity go up and up and up as the climate change is going into the future. So what do we see regionally at the lower emission scenario? And here we can see that we do expect to see some increased productivity. The darker green is the most productive, and so these are absolute values of stem volume. This doesn't change. When we look at the within program data set, it'll be changed. But this is absolute values of how much they're growing. And so, we have more productive areas down on the slopes and, some of the less productive areas really high up in the Cascades. But we can see that the more productive areas are getting bigger, even under the lower of the two emission scenarios. The emission scenario that is more likely is the higher one, and in this case you can see the high productivity areas increase even more across the region. And so just looking at this, you'd think, okay, well, this is great. We'll have more productive forests, and that's great. We can convert land, we can conserve species, we can have all these wonderful timber products, but the story starts to change when we remove the confounding effects of genetics and environment. And so this is at the plantation level, and you probably noticed right away that things aren't going up and up and up right now. We see a lot of programs that are just staying flat. And then when we get into the stronger emission scenario, we see some programs that are really decreasing. There are some areas in here where the sites are still increasing, and so there's a bit more variability. It's not one size fits all. Things are going up and down. So what happens when we take a look at what we might be able to see regionally in the future? So again, we're going to start with the lower of the two emission scenarios. And so because we're dealing with the within program data set, we can't look at the absolute values. We have to look at how much growth might change over time. So on both this slide and the next slide, the darkest blue color is areas that are actually seeing a decrease in growth. And so even on, in the lower emission scenario, you can start to see areas of decreased growth start to pop up here in southern Oregon, pop up here in the Olympic Peninsula, over here in the Cascades. So what happens when we move to the higher emission scenario? Well, that's a lot of area with decreased growth in 2085. So, these are models. There's a lot of uncertainty in these models. So, I wouldn't make land decisions based off this research and this research alone, but I do think that when I see this, it's something that I would be concerned about. And I think what it's also telling me is that while we can expect to see the conditions in our region to continue to be favorable for Douglas fir growth, it's not necessarily favorable for the populations that are currently growing there. And so it might be an argument for assisted migration and moving some of the populations around and really trying to match your seed source to your planting area. Just something to take in consideration. And if you wanted to take a look at this in detail for your sites, there are tools out there now to do that, and one of them is the seed lot selection tool available through the Forest Service and Oregon State University. Our conclusions for this project right now is that the relationships between the environmental characteristics and Douglas fir productivity are really subtle across the region. We are seeing precipitation as snow and annual heat moisture as being important for explaining variation in site productivity. And these models are predicting an increase in productivity across the landscape, but the projections might be biased upwards when we're looking at the across program data set because of the confounding genetic information in there. And the climate projections that we're using are also assuming very small changes in precipitation. So, if those climate projections turn out to be wrong, then how does that influence this model? And, like I said, there's still a lot of uncertainty in many of these things, but I think it's still interesting information. And so with that, I would like to acknowledge my mentor, Glenn Howe. He is the person who has funded and led all of these projects, and of course I finished my graduate work at Oregon State University. It was funded by the Pacific Northwest Tree Improvement Research Cooperative and the Center for Advanced Forestry Systems, and all of the data, all of the progeny test site data came from the Northwest Tree Improvement Cooperative, and that again is Keith J. Rickema and Terrence E. who put together some of the slides that I used today as well. So that's the end of my presentation. Thanks.